For years, researchers have worked toward transplanting animal kidneys into humans. It's a race against time. There are almost 600,000 Americans on dialysis in need of a life-saving kidney transplant. 240 Americans die every day on dialysis, and many of these deaths could be prevented if an unlimited supply of kidneys were available. Pigs are the most promising donor source. Researchers have been able to genetically engineer pig kidneys that can coexist with the human immune system. Surgeons have made multiple attempts to transplant animal organs into humans. But no one has been able to demonstrate how the entire process of transplant of a pig kidney to human would work in the real world. Until now. In seminal peer-reviewed research in the American Journal of Transplantation, a team at the University of Alabama at Birmingham revealed the results of the world's first clinical-grade pig-to-human kidney transplant. Notably, the study was conducted to meet the same standards as a Phase 1 clinical trial, making it mirror, as much as possible, every step of a normal transplant between humans. They started with unique pigs, genetically engineered with 10 gene edits. 3 deletions disabled genes that would trigger the human immune system and cause rejection. A 4th deletion prevents the kidneys from growing too large in the recipient after transplant. The rest are human genes added to the pig genome to prevent the recipient's immune system from attacking the organ. A herd of donor pigs live in a DPF, a designated pathogen-free facility designed for clinical use. The human recipient was a brain-dead man whose family gave permission for this crucial test of animal-to-human organ donation. Pig donor kidneys must be tissue-matched to human recipients, just like human kidneys. The UAB team designed and performed the world's first test to ensure tissue compatibility from a pig to a human. In separate tests, the researchers also confirmed there were no viruses transmitted. Both are critical to the wider implementation of pig-to-human kidney transplantation. After the pig's kidneys were removed, they were transported to UAB hospital, just like a human donor organ. Meanwhile, surgeons removed the recipient's kidneys. Then they attached the pig kidneys to the recipient's arteries and veins, as well as to the bladder. Within 23 minutes, the kidneys had produced urine. The kidneys remained viable and made urine for more than 3 days. And genetically modified pig cells were not detected in the recipient's blood. This study is an important step toward ending the organ shortage and preventing tens of thousands of deaths each year.